Hello there. Do you also have grandparents in your home who forgets that whether they have eaten, they have taken a nap, or whether who are you? That is really sad now. But do you know what is even more sad? That is, there are many young individuals who also suffer from the same problem. Just like the video that I showed you earlier, all these together correlates to a very fatal memory disorder, which is known as Alzheimer's disease. Now, talking about Alzheimer's, it marks its global presence by being the sixth most harmful disease in terms of mortality. And not only that, it destroys both the immediate as well as long-term memory formation. And now, there is exactly why I come in with my research, which deals with understanding a molecular mechanism of how this Alzheimer's work, which is even a mystery, even after like decades of research that has gone into. Oh, sorry to say, I am Arpan. Talking about Alzheimer's, it is formed because of a particular protein which is called amyloid beta. Now, amyloid beta is interesting because it is formed of a very unique class of proteins which are called intrinsically disordered proteins, which are very different from the normal concept of proteins that we always have. Why? Because IDPs don't have a structure associated. Now, this particular amyloid beta, if we think together, let's say we have a cell over here. Now, in this cell, as we can see, there is an intrinsically disordered protein precursor, which is the amyloid precursor protein. Now, it is a transmembrane protein that spans both the inside as well as outside the cell. But there are few agents in the cell which acts upon this, and then the daughter A beta is liberated from the mother APP. Now, when this daughter A beta is liberated in a concentration dependent manner, it can form these oligomers. Now, these oligomers are the major toxic species, not only from literature, but also mainly from our results that we have already shown earlier. But they don't stop over there. After forming these toxic structures, they form the final state of their aggregation, that is the fibrils, which are found in the post-mortem brains of the AD patients. Now, to understand simplistically about these oligomers, let me give you a demonstration. So with the aim of targeting amyloid beta, let's assume a pharma company designs a drug A, and it turns out that the drug is pretty effective against the monomers. But in our body, there are these toxic oligomers with different number and structure, to which the drug A might be completely ineffective. For that, only drug B can work. But how the company know a priori which drug to design? Or in other words, what is the number and structure of the oligomers? For looking at small objects like cells, we can take help of light microscopy. But if we want to detect even smaller objects like the oligomers, we have to take help from something known as fluorescence. But exactly there is the problem. Fluorescent molecules undergo bleaching when continuously exposed by light. But this is exactly what we have used to our advantage. So now, do you remember that initially there were some balls that were hurled on me? But do you know how many balls it were there? I'm sure you are not. But now let's say if those balls were hurled one by one, let's say like this. 1, 2, 3 and 4. Now you know that there were 4 balls that were being thrown on me. This is exactly how we have used bleaching of fluorescence to count the composition of an oligomer. To think of it pictorially, let us take a floodlight with some bulbs in it. To identify how many bulbs are there in the floodlight, we direct a heat gun on it which can break the bulbs. Now, if we monitor the floodlight intensity with time by keeping the heat gun pointed to it, we see the intensity decreases in steps as each bulb bursts. This is equivalent to a technique called single molecule photobleaching. Now in cells, the oligomers are similar to a floodlight as each A beta monomer contains a fluorescent tag. By using turf microscopy, we can see them separately and also bleach them. Counting these photobleaching steps gives us an idea of the nature of the oligomer. So, in conclusion, what we have found that by using our technique, there is a special tetramer among this group of amyloid beta oligomers which shows difference in its property, and this is also hinted in the literature which talks about its pore forming abilities. And nonetheless, by using our technique, we can actually screen multiple drugs against particular toxic oligomers of these amyloids.